I'm taking this the agar, the LB amp one, and pouring it into the glass bottle. Then I'm gonna take some water and pour it in and put it into the microwave.
And just toss these to the side. You don't have to record it every time, just record this once.
And then I'm pouring it into the little plates. I had a little difficulty making it even across the entire plate because I couldn't fill in some spots. But I eventually did. And I filled all the plates. So I started all the 5 plates.
Okay.
And it's taking a bit just to even it out. I did have enough to fill all 5 plates, so it was pretty good. Only filled the bottom to cover it fully. And then I'll repeat this process with the LB agar and not the M. And it's just a difference with the water, how much water you add, and I'm pouring that into the glass bottle and adding water and repeating the whole process and pouring it into 2 plates. I did mark the plates so I know which ones are which. This one, yeah, I spilled it a little on my finger, which was a little annoying, but it worked out. I just brushed it off my finger.
I spilled a little.
Okay.
And then it says use 500 50 milliliter, this one, cone of glass to, what's for measuring water? Tube for measuring.
I just went to go get water, and then I filled it up.
No, I mean 50, sorry.
150, yeah.
It's in the microwave, and then I took it out and put it into
Just so it covers the whole bottom, right? This one?
Both.
2 plates.
Okay, 1, 2, 3.
It's full for bacteria stuff.
Okay, should I just move it around? No, I don't want to. Dad, no.
So after that, I poured all of them. I poured the liquid in, and then I let all of them set. And then after they set, I came back and I took some of them. Yeah, this is not done yet. Okay, so I came back and I took some of the E. coli with the little loop, and I spread it across the LB amp. LB only, not amp, sorry. And I spread it across. After I was done spreading that one, I actually went in and did the other one because I had 2 plates. I think on the first one I did a better job spreading it.
But I'm
the next thing, after I let those set and the bacteria grew in the LB agar, it grew some bacteria, so I took that off the new loop, and then I took that and
the loop is filled.
Grabbed it and then put it into this liquid And I took some of the
To
liquid
tego, masz
and I
wszystko?
put it into that liquid there, and then
Poczekaj,
so
bo
like
widać.
basically bacteria, like food, the
Tutaj
bacteria. So I put that into, because there's bacteria and there's the bacteria, so I mixed the two bacteria and like the DNA and everything. So
Udałaś?
I then took some of this water that was at room temperature and mixed it with, and there was already powder inside this tube, so I mixed it with that. I did have a trouble shaking it, but I eventually did shake it till there wasn't any clumps or anything like that.
No, wystarczy, bo nie będzie miała potem miejsca, Gabi, żeby wymieszać. Close and shake the salty. No, pokaż tutaj, Gabi, musi być równo. 4 to 5 drops.
This isn't
Then
good
I
heating,
Hmm?
though. This one's not good.
took some of the liquid where I made the
But I'll choose
mixture
the best one.
of like the water and the agar and stuff like that, and I poured it onto the LB amp agar,
And just drop
and
it
then I spread it
on. A few
4
drops,
to
right?
5 drops and
after I was finishing adding a couple drops,
top of agar plate.
and about 4 drops.
A 2, a 3, a 4, a 5. Oh, you did do 1. I didn't do a
Then
1.
I took this little loop again and I spread it across.
Okay, and then I
Mix it
spread
up.
it. I streak it across, right?
Gently spread the bacteria around the plate.
Just basically spreading bacteria.
So there you can see my bottle of like Plas. So it was just lying there. But right now I'm just spreading this thing very aggressively. And then I left that for 24 hours. I flipped it upside down like you're supposed to. And then I don't know why I'm spreading it so aggressively. And then I took that, and then this was the result after 24 hours, and it did glow in the dark. It was pretty cool. So it's my